Hello everyone, today we're going to cover chapter 29, Development and Inheritance. So in this chapter, we will study the sequence of events from the fertilization of a secondary oocyte by a sperm cell to the formation of an adult organism. In particular, we will focus on the developmental sequence from fertilization through implantation, embryonic and fetal development, labor, and birth. We will also examine the principles of inheritance, which is the passage of hereditary traits from one generation to another. Development during pregnancy. Pregnancy is a sequence of events that begins with fertilization. It proceeds to implantation, then embryonic development, and then fetal development, and ideally it ends with birth about 38 weeks later or 40 weeks after the mother's last menstrual period. First, we're going to talk about the embryonic period. The embryonic period is the period from week 0 till week 8. First, we're going to talk about the first week of development. So the embryonic period, it extends from fertilization through the 8th week, right? The first week of development is characterized by several significant events, including fertilization, cleavage of the zygote, blastocyst formation, and implantation. So first, let's start with fertilization. So during fertilization, the genetic material from a haploid sperm cell, which is the spermatozoon, and a haploid secondary oocyte merges into a single diploid nucleus. So the way it happens is that of the 200 million sperm introduced into the vagina, fewer than 2 million, which is about only 1%, reach the cervix of the uterus, and only about 200 reach the secondary oocyte. So fertilization normally occurs in the uterine tube, which is the fallopian tube, within 12 to 24 hours after ovulation. Sperm can remain viable for about 48 hours after deposition in the vagina, although a secondary oocyte is viable for only about 24 hours after the ovulation. So thus pregnancy is most likely to occur if the intercourse it takes place during those 3-day window, like from 2 days before ovulation to 1 day after ovulation. So sperm swim from the vagina into the cervical canal by the whip-like movements of their tails, which is the flagella. The passage of sperm through the rest of the uterus and then into the uterine tube, it results mainly from contractions of walls of these organs. So the prostaglandins in semen are believed to stimulate uterine motility at the time of intercourse and to aid in the movement of sperm through the uterus and into the uterine tube. So sperm that reach the vicinity of the oocyte within minutes after ejaculation are not capable of fertilizing it until about 7 hours later. So during this time in the female reproductive tract, mostly in the uterine tube, sperm undergo capacitation. So capacitation is a series of functional changes that cause the sperm's tail to beat even more vigorously and prepare its plasma membrane to fuse with the oocyte's plasma membrane. So during capacitation, sperm are acted on by secretions in the female reproductive tract that result in the removal of cholesterol, glycoproteins, and proteins from the plasma membrane around the head of the sperm cell. So only capacitated sperm are capable of being attracted by and responding to those chemical factors produced by the surrounding cells of the ovulated oocyte. So for fertilization to occur, a sperm cell first must penetrate 2 layers. Okay, so the first layer is the corona radiata, the granulosa cells that surround the secondary oocyte, which we talked about it in the previous chapter. And the second layer is the zona pellucida, which is the clear glycoprotein layer between the corona radiata and the oocyte's plasma membrane. So the acrosome, which is a helmet-like structure that covers the head of a sperm, it contains several enzymes. Acrosomal enzymes and strong tail movements by the sperm help it penetrate the cells of the corona radiata and come in contact with the zona pellucida. So one of the glycoproteins in the zona pellucida acts as a sperm receptor. So its binding to specific membrane proteins in the sperm head triggers the acrosomal reaction, the release of the contents of the acrosome. So the acrosomal enzymes digest a path through the zona pellucida as the lashing sperm tail pushes the sperm cell onward. So although many sperm bind to glycoproteins in the zona pellucida and undergo acrosomal reactions, only that first sperm cell to penetrate the entire zona pellucida and reach the oocyte's plasma membrane, that's the one that fuses with the oocyte. So the fusion of a sperm cell with a secondary oocyte sets in motion events that block polyspermy. So polyspermy is fertilization by more than one sperm cell. So within a few seconds, the cell membrane of the oocyte depolarizes, which acts as a fast block to polyspermy. So fast block to polyspermy is the inability of a depolarized oocyte to fuse with another sperm. Depolarization also triggers the intracellular release of calcium ions, which stimulate exocytosis of secretory vesicles from the oocyte. The molecules released by exocytosis inactivate the glycoproteins in the zona pellucida and harden the entire zona pellucida. So those events are called the slow block to polyspermy. Once the sperm cell enters a secondary oocyte, the oocyte first must complete meiosis II. So it divides into a larger ovum, which is the mature egg, and a smaller second polar body that fragments and disintegrates. So the nucleus in the head of the sperm develops into the male pronucleus, and the nucleus of the fertilized ovum develops into the female pronucleus. So after the male and female pronuclei form, they fuse, producing a single diploid nucleus. So the fusion of the haploid N pronuclei restores the diploid number 2N, which is 46 chromosomes. The fertilized ovum now is called a zygote. So dizygotic twins, okay, we also call those fraternal twins. Those are produced from the independent release of two secondary oocytes and the subsequent fertilization of each by different sperm. They are the same age and in the uterus at the same time, but genetically they are as dissimilar as any other siblings. So dizygotic twins may or may not be the same sex. Also, we have the monozygotic twins, which is the identical twins. So because those monozygotic twins, they develop from a single fertilized ovum, they contain exactly the same genetic material and are always the same sex. Monozygotic twins arise from separation of the developing cells into two embryos, which in 99% of the cases occur before 8 days have passed. Separations that occur later than 8 days are likely to produce conjoined twins, a situation in which the twins are joined together and share some body structures. Okay, so here in this slide, we can see selected structures and events in fertilization. So here we have, so this is a sperm cell, and then also here we have the path of the sperm cell. So here outside layer, we have the corona radiata. We have the inside layer, which is the zona pellucida. And then also we have the plasma membrane of the secondary oocyte. And then here we have the cytoplasm of the secondary oocyte. And here we have the first polar body. This is figure A, sperm cell penetrating a secondary oocyte. And then here in figure B, we can see sperm cell in contact with a secondary oocyte with a scanning electron microscope. So here, as we can see, here we have a sperm cell, and then this is the acrosome, which is the head of the sperm cell, trying to penetrate to the secondary oocyte. And then here in this figure, we can see the male and the female pronuclei, which once they fuse together, they will make the zygote. Okay, so like we said, during fertilization, genetic material from a sperm cell and a secondary oocyte, they merge to form a single diploid nucleus, which is the zygote. So during fertilization, the genetic material from haploid sperm and haploid secondary oocyte, they merge into single diploid zygote. This happens in the uterine tube, which is the fallopian tube. Okay, so the sperm will undergo capacitation, which is changes brought about by the female reproductive tract that prepare the sperm for fertilization. And then the sperm must penetrate through two layers, and then we said the two layers are the corona radiata and the zona pellucida. Okay, and then the acrosomal enzymes and strong movements help with this process of penetration. Okay, and then also zona pellucida glycoproteins, they assist the sperm with this process. So that was the events for fertilization. Now we're going to talk about the cleavage of the zygote. So after fertilization, rapid mitotic cell divisions of the zygote called cleavage take place. The first division of the zygote begins about 24 hours after fertilization and is completed about 6 hours later. Each succeeding division takes slightly less time. So by the second day after fertilization, the second cleavage is completed and there are 4 cells. By the end of the third day, there are 16 cells. The progressively smaller cells produced by cleavage are called blastomeres. The successive cleavages eventually produce a solid sphere of cells called the morula. The morula is still surrounded by the zona pellucida and is about the same size as the original zygote. Next, we have blastocyst formation. So by the end of the fourth day, the number of cells in the morula increases as it continues to move through the uterine tube towards the uterine cavity. When the morula enters the uterine cavity on day 4 or day 5, a glycogen-rich secretion from the glands of the endometrium of the uterus passes into the uterine cavity and enters the morula through the zona pellucida. So this fluid called the uterine milk. So uterine milk, along with nutrients stored in the cytoplasm of the blastomeres of the morula, provides nourishment for the developing morula. At the 32-cell stage, the fluid enters the morula, collects between the blastomeres, and reorganizes them around a large fluid-filled cavity called the blastocyst cavity. The blastocyst cavity is also called the blastocele. Once the blastocyst cavity is formed, the developing mass is called the blastocyst. So though it now has hundreds and hundreds of cells, the blastocyst is still about the same size as the original zygote. During the formation of the blastocyst, 2 distinct cell populations arise. We have the embryoblast and the trophoblast. So the embryoblast, or the inner cell mass, is located internally and it eventually develops into the embryo. Okay, so that's the embryoblast. The other one is the trophoblast. So trophoblast is the outer superficial layer of cells that forms the sphere-like wall of the blastocyst. So it will ultimately develop into the outer chorionic sac that surrounds the fetus and the fetal portion of the placenta, the site of exchange of nutrients and wastes between the mother and the fetus. Around the 5th day after fertilization, the blastocyst hatches from the zona pellucida by digesting a hole in it with an enzyme and then squeezing through the hole. This shedding of the zona pellucida is necessary in order to permit the next step, which is the implantation or the attachment into the vascular, glandular, endometrial lining of the uterus. Cleavage and the formation of the morula and blastocyst. Okay, so here day 1, as we can see, here we have the polar bodies, And then here we have the blastomeres and we have the zona pellucida. Okay, so here as we can see, day 1 after fertilization, rapid mitotic cell divisions of the zygote called cleavage. So cleavage takes place. So the first division of the zygote, it begins about 24 hours after fertilization and is completed about 6 hours later. So as we can see here, we're going to see how each succeeding division is going to take less and less time. So here, day 2, so by the second day after fertilization, the second cleavage is completed and it has 4 cells. Okay, so as you can see here, we have 4 cells. And then day 4, or by the end of the 3rd day, there are 16 cells. And so they progressively smaller cells produced by cleavage, those are called blastomeres. So the successive cleavages, eventually they produce a solid sphere of cells called the morula. Okay, so those are going to be called the morula. And then the morula is still surrounded by the zona pellucida. Okay, so here, this is going to be the zona pellucida, and then is about the same size as the original zygote. So day 5, here figure D. So at the 32 cell stage, okay, so here we have the 32 cells. The fluid enters the morula. It collects between the blastomeres and reorganizes them around a large fluid filled cavity. This is going to be called the blastocyst cavity. And then here, if we can see here, like on day 5, here inside we have the this cavity here is going to be the blastocyst cavity. So once the blastocyst cavity is formed, the developing mass is called the blastocyst. Okay, so here this whole thing is going to be the blastocyst. So now it has hundreds of cells. The blastocyst is still going to be the same size as the original zygote, or yes, the original zygote. So during the formation of the blastocyst, we have 2 distinct cell populations that arise. So we have the embryoblast, right? This is the first one. And then the second one was the trophoblast. So like we said, the embryoblast is going to be the inner, okay, inner cell mass. So here, okay, those cells inside. They're located internally and eventually they develop into the embryo. And as for the trophoblast, okay, so it's going to be the outer superficial layer of the cells, okay, and then they form the sphere-like wall of the blastocyst. So, and then as we said, around the 5th day after the fertilization, so the blastocyst is going to hatch from the zona pellucida by digesting a hole in it with an enzyme and then squeezing through the hole. Next we have implantation. The blastocyst remains free within the uterine cavity for about 2 days before it attaches to the uterine wall. At this time, the endometrium is in its secretory phase. About 6 days after fertilization, the blastocyst loosely attaches to the endometrium in a process called implantation. So here, as we can see in this figure, here we have the endometrium of the uterus, and then here we have the blastocyst, and then we can see how the blastocyst loosely attaches to the endometrium, and this is called implantation. So as the blastocyst implants, usually in either the posterior portion of the fundus or the body of the uterus, it orients with the inner cell mass towards the endometrium. So about 7 days after fertilization, the blastocyst attaches to the endometrium more firmly, as we can see here in this figure. Endometrial glands in the vicinity enlarges. Okay, so here we had the endometrial glands, and then in this figure we can see how those endometrial glands in the vicinity, they enlarge, and the endometrium becomes more vascularized, and then here we can see how we have more blood vessels. The blastocyst eventually secretes enzymes and burrows in the endometrium and becomes surrounded by it. So the following implantation, the endometrium is known as the decidua. Decidua means falling off. Okay, so the decidua separates from the endometrium after the fetus is delivered, much as it does in normal menstruation. Okay, so also just like we talked about in the previous slide, so here we have the trophoblast, and then here we have the embryoblast, right? So the embryoblast was the inner cell mass, okay, and then the trophoblast was the outer superficial layer of the cells, and then those are the ones that form the sphere-like wall of the blastocyst. The major events associated with the first week of development are summarized in this figure. And as we can see, we have the first one is fertilization. And then as we said, it occurs within the uterine tube 12 to 24 hours after ovulation, right? So here we have ovulation taking place. And then here in the uterine tube, that's where fertilization takes place. And then here we have the different stages of cleavage, like 2 cells, 4 cells, 16 cells, 32 cells, right? And then eventually we have here morula, 3-4 days after fertilization. And then here we have the blastocyst formation. And then finally we have implantation. Now we're going to talk about the second week of development, week 2, which is the development of the trophoblast. A quick reminder from the previous slide, the trophoblast is the outer superficial layer of cells that forms the sphere-like wall of the blastocyst. Okay, so during the second week of development, about 8 days after fertilization, the trophoblast develops into 2 layers in the region of contact between the blastocyst and the endometrium. So these are a syncytiotrophoblast that contains no distinct cell boundaries and a cytotrophoblast between the embryoblast and the syncytioblast that is composed of distinct cells. The 2 layers of trophoblast become part of the choroid. Choroid is one of the fetal membranes as they undergo further growth. During implantation, the syncytiotrophoblast secretes enzymes that enable the blastocyst to penetrate the uterine lining by digesting and liquefying the endometrial cells. Eventually, the blastocyst becomes buried in the endometrium and inner 1/3 of the myometrium. Another secretion of the trophoblast is the human chorionic gonadotrophin hormone, the HCG, which has action similar to LH. So HCG rescues the corpus luteum from degeneration and sustains its secretion of progesterone and secretion of estrogens. These hormones, they maintain the uterine lining in a secretory state, hence preventing the process of menstruation. So the peak secretion of HCG, it occurs about the 9th week of pregnancy, at which time the placenta is fully developed and produces the progesterone and estrogens that continue to sustain the pregnancy. So usually the presence of HCG in maternal blood or in the urine is an indicator of pregnancy and is detected by the home pregnancy tests. The development of the bilaminar embryonic disc. Like those of the trophoblasts, cells of the embryoblast also differentiate into 2 layers around 8 days after fertilization. So we have a hypoblast and the epiblast. The hypoblast, also called primitive endoderm, and the epiblast, also called as primitive ectoderm. So cells of the hypoblast and the epiblast together, they form a flat disc referred to as the bilaminar embryonic disc. Soon, a small cavity appears within the epiblast and eventually enlarges to form the amniotic cavity. Next, we have the development of the amnion. So as the amniotic cavity enlarges, a single layer of squamous cells forms a dome-like roof above the epiblast cells called the amnion, which we're going to see it in the figure in a couple of slides. Thus, the amnion forms the roof of the amniotic cavity, and the epiblast forms the floor. Initially, the amnion overlies only the bilaminar embryonic disc. However, as the embryonic disc increases in size and begins to fold, the amnion eventually surrounds the entire embryo, creating the amniotic cavity that becomes filled with the amniotic fluid. So most amniotic fluid is initially derived from maternal blood. So later, the fetus contributes to the fluid by excreting urine into the amniotic cavity. So amniotic fluid serves as a shock absorber for the fetus, helps regulate fetal body temperature, it helps prevent the fetus from drying out and prevents adhesions between the skin of the fetus and surrounding tissues. The amnion usually ruptures just before birth. It and its fluid constitute the bag of waters. You know when they say my water broke, right? So that's what it means. Embryonic cells are normally stuffed off into amniotic fluid. They can be examined in a procedure called amniocentesis, which involves withdrawing some of the amniotic fluid that bathes the developing fetus and analyzing the fetal cells and the dissolved substances. Development of the yolk sac. Also, on the 8th day after fertilization, the cells at the edge of the hypoblast, they migrate and cover the inner surface of the blastocyst wall. So the migrating columnar cells become squamous, it becomes flat, and then form a thin membrane referred to as the exocoelomic membrane. Together with the hypoblast and the exocoelomic membrane forms the wall of the yolk sac, the former blastocyst cavity during earlier development. So as a result, the bilaminar embryonic disc is now positioned between the amniotic cavity and the yolk sac, which we're going to see it in the figure in a couple of slides. Since human embryos receive their nutrients from the endometrium, the yolk sac is relatively empty and small and decreases in size as the development progresses. So nevertheless, the yolk sac has several important functions in humans. So the yolk sac, it supplies nutrients to the embryo during 2nd and 3rd weeks of development. The yolk sac is the source of blood cells from the 3rd through 6th weeks. It contains the first cells that will eventually migrate into the developing gonads, differentiate into the primitive germ cells, and many more. Now we're going to talk about the development of sinusoids, the development of extraembryonic mesoderm, and the development of choroid. So first, let's talk about the development of sinusoids. So on the 9th day after fertilization, the blastocyst becomes completely embedded in the endometrium. So as the syncytiotrophoblast expands, small spaces called lacunae develop within it. So by the 12th day of development, the lacuna fuse to form larger interconnecting spaces called lacunar networks. Endometrial capillaries around the developing embryo become dilated and are referred to as maternal sinusoids. As the syncytiotrophoblast erodes some of the maternal sinusoids and endometrial glands, maternal blood and secretions from the glands enter the lacunar networks and flow through them. Maternal blood is both a rich source of materials for embryonic nutrition and a disposal site for the embryo's wastes. Now we're going to talk about the development of the extraembryonic mesoderm. So about the 12th day after fertilization, the extraembryonic mesoderm develops. These mesodermal cells, they are derived from the yolk sac and they form a connective tissue layer, which we call those mesenchyme, around the amnion and the yolk sac. Soon, a number of large cavities, they develop in the extraembryonic mesoderm, which then fuse to form a single larger cavity called the extraembryonic coelom. And finally, we have the development of the chorion. So the extraembryonic mesoderm together with the two layers of the trophoblast, remember the trophoblast had the cytotrophoblast and the syncytiotrophoblast. Okay, so together with the two layers of the trophoblast forms the chorion. The chorion surrounds the embryo and later the fetus. Eventually, it becomes the principal embryonic part of the placenta, the structure for exchange of materials between mother and fetus. The chorion also protects the embryo and fetus from the immune responses of the mother in two ways. It secretes proteins that block antibody production by the mother. Second, it promotes the production of T lymphocytes that suppress the normal immune response in the uterus. Finally, the chorion produces human chorionic gonadotropin, the HCG, an important hormone for pregnancy. The inner layer of the chorion eventually fuses with the amnion. With the development of the chorion, the extraembryonic coelom is now referred to as the chorionic cavity. By the end of the second week of development, the bilaminar embryonic disc, it becomes connected to the trophoblast by a band of extraembryonic mesoderm called the connective stalk, okay, or connecting body. The connecting stalk will become the future umbilical cord. Okay, so let's go over some of the stuff that we talked about in the previous slides by looking at this figure. Okay, so this figure shows us principal events of the second week of development. About 8 days after fertilization, the trophoblast develops into a syncytiotrophoblast and a cytotrophoblast. The embryoblast develops into a hypoblast and epiblast, which is the bilaminar embryonic disc. Okay, so here in this figure, we can see the frontal section through endometrium of uterus showing blastocyst about 8 days after fertilization. Okay, so here, this section here, we have the endometrium of the uterus. Here, this is the endometrial gland. This whole thing is the blastocyst, right? Here inside, we have the blastocyst cavity. Okay, so here we have the trophoblast, and then like we said, the two layers of the trophoblast is made up of the cytotrophoblast, and the outside here we have the syncytiotrophoblast. And then here, as we can see, here we have the amnion, and then inside here we have the amniotic cavity, the blood vessels, and then here we have the bilaminar embryonic disc, which is made up of the hypoblast and the epiblast. So here the inner one is going to be the epiblast, and then here we have the hypoblast, and then finally we have the formation of exocoelomic membrane. Okay, so I'm gonna go over the development stages of the second week of development again. So at first we have the trophoblast, right? The development of the trophoblast. So like we said, about 8 days after fertilization, the trophoblast is gonna develop into two layers in the region of contact between the blastocyst and the endometrium. And then as we can see here in this figure, right, we have the syncytiotrophoblast and the cytotrophoblast. Okay, so like we said, the syncytiotrophoblast is gonna contains no distinct cell boundaries, and the cytotrophoblast is between the embryoblast and the syncytiotrophoblast that is composed of distinct cells. So the two layers of trophoblast become part of the chorion as they undergo further growth. So during implantation, the syncytiotrophoblast secretes enzymes that enable the blastocyst to penetrate the uterine lining by digesting and liquefying the endometrial cells. So eventually, as we can see, the blastocyst, it becomes buried in the endometrium and inner 1/3 of the myometrium. So second, here we have the development of the bilaminar embryonic disc. So so like those of the trophoblasts, cells of the embryoblast also differentiate into two layers around 8 days after fertilization. Okay, so we have the hypoblast and the epiblast. So cells of the hypoblast and the epiblast together, they form the flat disc referred to as the bilaminar embryonic disc. And then, like we said, soon a small cavity appears within the epiblast and eventually it enlarges, and then it makes the number 3, the amniotic cavity. So third, we have the development of the amnion. So like we said, as the amniotic cavity enlarges, a single layer of squamous cells forms a dome-like roof above the epiblast, and then we call those amnion. So the amnion forms the roof of the amniotic cavity, and the epiblast, it forms the floor. And then finally, we have the development of the yolk sac. Okay, so here, as we can see, also on the 8th day of fertilization, cells at the edge of the hypoblast, they're going to migrate and cover the inner surface of the blastocyst wall. So the migrating columnar cells, they become squamous, flat, as we can see in this figure. Okay, and then form a thin membrane that we call it the exocoelomic membrane. Okay, just like we can see here in this figure. So here, if we look closer in this figure is showing us the frontal section through endometrium of uterus showing blastocyst about 9 days after fertilization. Okay, so here we have the endometrium of the uterus. Here we have the blood vessels. And then we have here the lacuna, right? And then here we have the trophoblast, which is made up of the cytotrophoblast and the syncytiotrophoblast, okay, this region here. And then also we have the those flat squamous cells are gonna make the exocoelomic membrane. Here, this is where we have the development of the yolk sac, right? And then here we have the bilaminar embryonic disc, which is made up of the epiblast and the hypoblast. So here we have the epiblast in blue and the hypoblast in yellow. And then this cavity here is gonna be the amniotic cavity. Okay, so this is the stage where we have the development of the yolk sac. So like we said, together with the hypoblast, the exocoelomic membrane forms the wall of the yolk sac, the former blastocyst cavity during earlier development. So as a result, the bilaminar embryonic disc is now positioned between the amniotic cavity and the yolk sac. As for the development of sinusoids, the development of the extraembryonic coelom, and the development of the choroid, as you can see in this figure. So here we have a frontal section through endometrium of uterus showing blastocyst about 12 days after fertilization. Okay, so first the development of the sinusoids. So by the 12th day of development, the lacunae, okay, so those are the lacunae, okay, fuse to form larger interconnecting spaces called the lacunar network. And then you see how here they fuse together and they make a larger lacunar network. Endometrial capillaries around the developing embryo become dilated and are referred to as the maternal sinusoids. Okay, so here we have the maternal sinusoids here. And then as for the development of the extra embryonic coelom, so about the 12th day after fertilization, the extra embryonic mesoderm, it develops. Okay, so here this part is going to be the extra embryonic mesoderm develops. These mesodermal cells are derived from the yolk sac. So here we have the yolk sac. Okay, and then form a connective tissue layer, mesenchyme, around the amnion and the yolk sac. Here we have the amnion, here we have the yolk sac, and then here we have the mesenchyme. And then soon a number of large cavities, they develop in the extraembryonic mesoderm, which then they fuse together to form a single larger cavity called the extraembryonic coelom. Okay, so here, and then now we're gonna talk about the development of the chorion. Okay, so here we have the development of the chorion. So as we know, the extraembryonic mesoderm here, together with the two layers of the trophoblast, which is the cytotrophoblast and the syncytiotrophoblast, they form the chorion. Okay, and then here it's gonna be this part here. Now we're gonna talk about the third week of development. So the third embryonic week begins a 6-week period of very rapid development and differentiation. So during the third week, the 3 primary germ layers are established and lay the groundwork for organ development in weeks 4 through 8. Gastrulation. The first major event of the third week of development is the gastrulation. It occurs about 15 days after fertilization. So in this process, the bilaminar embryonic disc consisting of the epiblast and the hypoblast transforms into a trilaminar embryonic disc consisting of the 3 layers. Okay, so here the bilaminar embryonic disc, which has 2 layers, right, transforms into a trilaminar embryonic disc to a 3-layered, consisting of those 3 layers, which are the ectoderm, and then the mesoderm, and the endoderm. So these primary germ layers are the major embryonic tissues from which the various tissues and organs of the body develop. Gastrulation involves the rearrangement and migration of cells from the epiblast. So the first evidence of gastrulation is the formation of the primitive streak, a faint groove on the dorsal surface of the epiblast that elongates from the posterior to the anterior part of the embryo. The primitive streak, okay, so the primitive streak clearly establishes the head and tail ends of the embryo, as well as its right and left sides. At the head end of the primitive streak, a small group of epiblastic cells forms a rounded structure called the primitive node. Okay, so there is the primitive node. Following formation of the primitive streak, cells of the epiblast move inward below the primitive streak and detach from the epiblast. So this process is called invagination. So once the cells have invaginated, some of them displace the hypoblast, forming the endoderm. Other cells remain between the epiblast and newly formed endoderm to form the mesoderm, which is the middle layer. And then cells remaining in the epiblast then form the ectoderm, which is the outside layer. So the ectoderm and the endoderm are epithelia composed of tightly packed cells. The mesoderm is a loosely organized connective tissue, which we call it the mesenchyme. Okay, and then as the embryo develops, the endoderm ultimately becomes the epithelial lining of the gastrointestinal tract, the respiratory tract, and several other organs. The mesoderm gives rise to muscles, gives rise to bones and other connective tissues, and the peritoneum. And then as for the ectoderm develops into the epidermis of the skin and the nervous system. So here we have a quote by Louis Wolpert from 1986, says it is not birth, marriage, or death, but gastrulation, which is truly the most important time in your life. So here this figure explains gastrulation, just like we talked about it in the previous slide. So gastrulation involves the rearrangement and migration of cells from the epiblast. So here in this figure, it's showing us a transverse section of trilaminar embryonic disc about 16 days after fertilization. So in the process of gastrulation, like we said, the bilaminar, the 2-layered embryonic disc consisting of the epiblast and the hypoblast, right? So here is going to transform into trilaminar embryonic disc, as we can see here, okay? And then it's going to make the 3-layer, the primary germ layers, which is going to be the ectoderm, the endoderm, and the mesoderm. Okay, those 3 layers. And then, like we said, these primary germ layers, those are going to be the major embryonic tissues from which the various tissues and organs of the body are going to develop. Okay, also we said that gastrulation involves the rearrangement and migration of cells from the epiblast. Okay, so here the first evidence of gastrulation is going to be the formation of the primitive streak. Okay, so here, and then which is a faint groove on the dorsal surface of the epiblast that elongates from the posterior to the anterior part of the embryo, as we can see here in this figure. Okay, so also, oh, by the way, here, this part is going to be the dorsal and partial sectional views of bilaminar embryonic disc about 15 days after fertilization. And then this is going to be the transverse section of trilaminar embryonic disc about 16 days after fertilization. Okay, so just like we said, it makes the primitive streak. Okay, and then the primitive streak clearly establishes the head and the tail. Okay, so the head end, and then we have the tail end of the embryo, as well as its right and left side. At the head end of the primitive streak, a small group of epiblastic cells forms around the structure called the primitive node, right? So here we have the primitive node. Following formation of the primitive streak, cells of the epiblast, they're going to move inward below the primitive streak and detach from the epiblast, and then this is called invagination. Okay, so here, as you can see, this process is going to be invagination. So once the cells have invaginated, some of them displace the hypoblast, they form the endoderm. Okay, so here you have the endoderm. So other cells, they remain between the epiblast and the newly formed endoderm to form the mesoderm. Okay, so here, that layer there, and then cells remaining in the epiblast then form the ectoderm, which is the outer layer. So about 16 days after fertilization, mesodermal cells from the primitive node, they migrate toward the head end of the embryo and form a hollow tube of cells in the midline called the notochordal process. So by days 22 to 24, this notochordal process becomes a solid cylinder of cells called the notochord. Okay? So here we have the notochord. The notochord includes certain mesodermal cells to develop into the vertebral bodies. It also forms the nucleus pulposus of the intervertebral discs. Also, during the 3rd week of development, 2 faint depressions, okay, so 2 faint depressions appear on the dorsal surface of the embryo where the ectoderm and the endoderm make contact but lack mesoderm between them. The structure closer to the head end is called the oropharyngeal membrane, okay, and which is pertaining to the pharynx, right? It breaks down during the 4th week to connect the mouth cavity to the pharynx and the remainder of the gastrointestinal tract. The structure closer to the tail end is going to be called the cloacal membrane, okay, which degenerates in the 7th week to form the openings of the anus and the urinary and reproductive tracts. When the cloacal membrane appears, the wall of the yolk sac forms a small vascularized outpouching called the allantois, okay, so that extends into the connecting stalk. In non-mammalian organisms enclosed in an amnion, the allantois is used for gas exchange and waste removal. Because of the role of the human placenta in these activities, the allantois is not a prominent structure in humans. Nevertheless, it does function in the early formation of blood and blood vessels and is associated with the development of the urinary bladder. Okay, so here let's go over the development of the notochordal process by looking at this figure here. So like we said, the notochordal process, it develops from the primitive node and later becomes the notochord. So here in this figure, it's showing us the A is dorsal and partial sectional views of trilaminar embryonic disc about 16 days after fertilization. And then here we have the sagittal section of trilaminar embryonic disc about 16 days after fertilization. Okay, so basically it's the same figure but different sectional views. Okay, so about 16 days after fertilization, like we said, the mesodermal cells from the primitive node, they migrate toward the head end of the embryo and form a hollow tube of cells in the midline called the notochordal process. Okay, so here we have the notochordal process here, and then another one here in the sagittal section view. Okay, so and then as we said, by days 22 to 24, the notochordal process becomes a solid cylinder of cells called the notochord. Okay, and then the notochord, it induces certain mesodermal cells, okay, and then to develop into the vertebral bodies, okay? And then here we have the mesoderm. Also, during the third week of development, we said we have two faint depressions that appear on the dorsal surface of the embryo, where the ectoderm and the endoderm, they make contact but lack the mesoderm between them. Okay, and then the structure closer to the head end is going to be the oropharyngeal membrane. Okay, so here this is going to be the oropharyngeal membrane. And then here on this figure, this part is going to be the oropharyngeal membrane. And then it breaks down during the fourth week to connect the mouth cavity to the pharynx and the remainder of the gastrointestinal tract. And then also we have the structure that is closer to the tail end. Okay, it's gonna be the cloacal membrane, okay, here. And then let's see if we can see the cloacal membrane on this figure. Yes, it's gonna be here on the tail end. Okay, so head and tail end, and then here we have the cloacal membrane. Okay, so just like we said, it's gonna degenerate in the 7th week, and then it's gonna form the openings of the anus and urinary and reproductive tracts. And then, so when the cloacal membrane appears, the wall of the yolk sac forms a small vascularized pouching called the allantois. So here we have the connecting stalk, and then here we have the allantois on that figure. Here we can only see the connecting stalk in this figure. Okay, and then so allantois is gonna extend into the connecting stalk. Now we're gonna talk about neurulation. So in addition to inducing mesodermal cells to develop into vertebral bodies, the notochord also induces ectodermal cells over it to form the neural plate. Okay, to form the neural plate. By the end of the 3rd week, the lateral edges of the neural plate become more elevated and form the neural fold. The depressed mid-region is called the neural groove. Generally, the neural folds approach each other and fuse, thus converting the neural plate into a neural tube. Okay, and then this occurs first near the middle of the embryo, and then it progresses towards the head and tail ends. Neural tube cells then develop into the brain, and they develop into the spinal cord. As the neural tube forms, some of the ectodermal cells form the tube, migrate to form several layers of cells called the neural crest. Here we have the neural crest. Neural crest cells, they give rise to all sensory neurons and postganglionic neurons of the peripheral nerves, the adrenal medulla, melanocytes of the skin, arachnoid matter and pia matter of the brain and spinal cord, and almost all of the skeletal and connective tissue components of the head. At about 4 weeks after fertilization, the head end of the neural tube develops into 3 enlarged areas called primary brain vesicles. Okay, so here we have the 3 primary brain vesicles. So we have the prosencephalon, or the forebrain. Okay, we have the mesencephalon, or the midbrain, and then finally we have the rhobencephalon, or the hindbrain. So by the end of the 2nd week of development, chorionic villi begin to develop. So those are these finger-like projections. They consist of chorion that projects into the endometrial wall of the uterus. So by the end of the 3rd week, blood capillaries develop in the chorionic villi. Blood vessels in the chorionic villi, they connect to the embryonic heart by way of the umbilical arteries and umbilical vein through the connecting stalk, which will eventually become the umbilical cord. The fetal blood capillaries within the chorionic villi, they project into the lacunae, which unite to form the intravillous spaces that bathe the chorionic villi with maternal blood. So as a result, the maternal blood bathes the chorion-covered fetal blood vessels. Note, however, that the maternal and the fetal blood vessels, they don't join, and the blood they carry does not normally mix. Instead, oxygen and nutrients in the blood of the mother's intravillous spaces, the spaces between chorionic villi, diffuse across the cell membranes into the capillaries of the villi. And as for the waste products such as carbon dioxide, those diffuse in the opposite direction. So the 4th through 8th weeks of development are very significant in embryonic development because all major organs, they appear during this time. The term organogenesis, it refers to the formation of body organs and systems. So by the end of the 8th week, all of the major body systems have begun to develop, although their functions for the most part are minimal. Organogenesis requires the presence of blood vessels to supply developing organs with oxygen and other nutrients. So during the 4th week after fertilization, the embryo undergoes very dramatic changes in shape and size, nearly tripling its size. It is essentially converted from a flat, 2-dimensional trilaminar embryonic disc to a 3-dimensional cylinder, a process called embryonic folding. So also the GI tract, it begins to form. And then during the 5th week of development, there is a very rapid development of the brain. So growth of the head is going to be considerable. By the end of the 6th week, the head grows even larger relative to the trunk, and the limbs show substantial development. So in addition, the neck and the trunk, they begin to straighten, and the heart is now 4-chambered. So by the 7th week, the various regions of the limbs become distinct and the beginnings of the digits appear. So now, at the start of the 8th week, which is the final week of embryonic period, the digits of the hands are short and webbed, the tail is shorter but still visible, the eyes are open, and the auricles of the ears are visible. So by the end of the 8th week, all regions of limbs are apparent, the digits are distinct and no longer webbed due to removal of cells via apoptosis. Apoptosis is a programmed cell death. Also, the eyelids come together and may fuse, the tail disappears, and the external genitals begin to differentiate. So now the embryo has clearly human characteristics. Now we're going to talk about placentation. So placentation is the process of forming the placenta, the site of exchange of nutrients and wastes between the mother and the fetus. The placenta also produces hormones needed to sustain the pregnancy. The placenta is unique because it develops from two separate individuals, the mother and the fetus. So by the beginning of the 12th week, the placenta has two distinct parts. It has the fetal portion and it has the maternal portion, okay? So the fetal portion is formed by the chorionic villi of the chorion, and the maternal portion is formed by the decidua bacillus of the endometrium. So when fully developed, the placenta is shaped like a pancake. Functionally, the placenta allows oxygen and nutrients to diffuse from maternal blood into fetal blood, while carbon dioxide and wastes diffuse from fetal blood into maternal blood. The placenta also is a protective barrier because most microorganisms, they cannot pass through the placenta. However, there are certain viruses such as those that cause AIDS, the German measles, chickenpox, measles, encephalitis, and others, those can actually cross the placenta. Also, many drugs, alcohol, and some substances that can cause birth defects, those also can pass truly through the placenta. The placenta stores nutrients such as carbohydrates, proteins, calcium, and iron, which are released into fetal circulation as required. The actual connection between the placenta and embryo and later the fetus is through the umbilical cord, which develops from the connecting stalk, just like we said in the previous slides. The umbilical cord, it consists of two umbilical arteries that carry the oxygenated fetal blood to the placenta. One umbilical vein that carries oxygen and nutrients acquired from the mother's intervillous spaces into the fetus. So in some cases, the umbilical vein is used to transfuse blood into a fetus or to introduce drugs for various medical treatments. So after the birth of the baby, the placenta detaches from the uterus and is therefore termed the afterbirth. So at this time, the umbilical cord is tied off and then severed. The small portion, about an inch of the cord that remains attached to the infant, as we all have seen that, right, begins to wither and eventually it falls off, okay? Usually it falls off within 12 to 15 days after birth. So the area where the cord was attached, it becomes covered by a thin layer of skin and scar tissue forms. So the scar is going to be the umbilicus or the navel. Fetal period 9 weeks and through 30 weeks. So during the fetal period, which is from 9th week until birth, tissues and organs that develop during the embryonic period grow and they differentiate. Very few new structures appear during the fetal period, but the rate of body growth is remarkable, especially during the second half of intrauterine life. For example, during the last 2.5 months of intrauterine life, half of the full term weight is added. At the beginning of the fetal period, the head is half the length of the body. By the end of the fetal period, the head size is only 1/4 the length of the body. So during the same period, the limbs also increase in size from 1/8 to 1/2 the fetal length. The fetus is also less vulnerable to the damaging effects of drugs, radiation, microbes than it was when it was only an embryo. So here this figure is a summary of the major developmental events of the embryonic and fetal periods. So here, as we can see, this figure is a summary of representative developmental events of the embryonic and fetal periods. The embryos and fetuses are not shown at their actual sizes, of course. So here we can see development during the fetal period is mostly concerned with the growth and differentiation of tissues and organs formed during the embryonic period. And then here we can see how during the 20-day embryo, 24-day embryo, 32-day embryo, 44-day embryo, 52-day embryo. Here we have 10 weeks fetus, 13-week fetus, and then finally the 26th fetus. Maternal changes during pregnancy. Hormones of pregnancy. During the first 3 to 4 months of pregnancy, the corpus luteum in the ovary continues to secrete progesterone and estrogens, which maintain the lining of the uterus during pregnancy and prepare the mammary glands to secrete milk. The amounts secreted by the corpus luteum, however, are only slightly more than those produced after ovulation in a normal menstrual cycle. From the 3rd month through the remainder of the pregnancy, the placenta itself provides the high levels of progesterone and estrogens that are required. So as noted previously, the chorion of the placenta secretes the human chorionic gonadotropin, the HCG, into the blood. So in turn, HCG stimulates the corpus luteum to continue production of progesterone and estrogens, which is an activity required to prevent menstruation and for the continued attachment of the embryo and fetus to the lining of the uterus. So by the 8th day after fertilization, HCG can be detected in the blood and urine of a pregnant woman. And then peak secretion of HCG occurs at about the 9th week of pregnancy. During the 4th and 5th months, the HCG level decreases sharply and then levels off until childbirth. The chorion begins to secrete estrogens after the first 3 or 4 weeks of pregnancy and progesterone by the 6th week. These hormones are secreted in increasing quantities until the time of birth. So by the 4th month, when the placenta is fully established, the secretion of HCG is greatly reduced and the secretions of the corpus luteum are no longer essential. A high level of progesterone ensures that the uterine myometrium is relaxed and that the cervix is tightly closed. After delivery, estrogens and progesterone in the blood decrease to normal levels. Relaxin is a hormone produced first by the corpus luteum of the ovary and later is produced by the placenta. It increases the flexibility of the pubic symphysis and it helps dilate the uterine cervix during labor. So both of these actions, they ease the delivery of the baby. Adjustments of infant at birth. So during pregnancy, the embryo and later the fetus is totally dependent on the mother for its existence. The mother supplies the fetus with oxygen, it supplies the fetus with nutrients, eliminates its carbon dioxide, eliminates other waste, it protects it against shocks and temperature changes, and it provides antibodies that confer protection against certain harmful microbes. At birth, a physiologically mature baby becomes much more self-supporting and the newborn's body systems must make various adjustments. The most dramatic changes occur in the respiratory system and the cardiovascular system. So first, let's look at the respiratory system adjustments. Okay, so the reason that the fetus depends entirely on the mother for obtaining oxygen and eliminating carbon dioxide is that the fetal lungs are either collapsed or partially filled with amniotic fluid. The production of surfactant begins by the end of the 6th month of development. Because the respiratory system is fairly well developed at least 2 months before birth, premature babies delivered at 7 months are able to breathe and cry. After delivery, the baby's supply of oxygen from the mother ceases and any amniotic fluid in the fetal lungs is observed. Because carbon dioxide is no longer being removed, it's going to start building up in the blood. So a rising carbon dioxide level is going to stimulate the respiratory center in the medulla oblongata, causing the respiratory muscles to contract and the baby to draw his or her first breath. Because the first inspiration is unusually deep, as the lungs contain no air, the baby also exhales vigorously and naturally starts crying. A full-term baby may breathe 45 times a minute for the first 2 weeks after birth, and the breathing rate gradually will decline until it approaches a normal rate of 12 breaths per minute. As for the cardiovascular system adjustments, okay, so after the baby's first inspiration, the cardiovascular system must make several adjustments. Closure of the foramen ovale between the atria of the fetal heart, which occurs at the moment of birth, diverts the oxygenated blood to the lungs for the first time. The umbilical arteries and umbilical veins become ligaments. The milk ejection reflex. Oxytocin causes release of milk into the mammary ducts via the milk ejection reflex. Milk formed by the glandular cells of the breast is stored until the baby begins active suckling. Stimulation of touch receptors in the nipple, it initiates sensory nerve impulses that are relayed to the hypothalamus. In response, secretion of oxytocin from the posterior pituitary increases. Carried by the bloodstream to the mammary glands, oxytocin, it stimulates contraction of myoepithelial cells, which are the smooth muscle-like cells surrounding the glandular cells and ducts. The resulting compression moves the milk from the alveoli of the mammary glands into the mammary ducts, where it can be suckled by the baby. This process is termed as milk ejection. It means milk let down. Even though the actual ejection of milk does not occur until 30 to 60 seconds after the nursing begins, the latent period, some milk stored in lactiferous sinuses near the nipple is available during the latent period. Stimuli other than suckling, such as hearing a baby's cry or touching the mother's genitals, also can trigger oxytocin release and milk ejection. During late pregnancy and the first few days after birth, the mammary glands secrete a cloudy fluid called the colostrum. Although it is not as nutritious as milk, it contains less lactose and virtually no fat. So colostrum serves adequately until the appearance of true milk on about the 4th day. Colostrum and maternal milk contain important antibodies that protect the infant during the first few months of life. Also, colostrum is rich in carbohydrates, it's rich in proteins and antibodies, low in fat. Fats in hard to digest because fats are hard to digest for newborns. And then concentrated nutrients well suited for small digestive system of newborns. Lactation often blocks ovarian cycles for the first few months following delivery if the frequency of suckling is about 8 to 10 times a day. This effect is inconsistent. However, an ovulation commonly precedes the first menstrual period after delivery of a baby. So as a result, the mother can never be certain if she is not fertile. Breastfeeding is therefore an unreliable birth control measure. A primary benefit of breastfeeding is nutritional. Human milk is a sterile solution that contains amounts of fatty acids, lactose, amino acids, minerals, vitamins, and water that are ideal for the baby's digestion, for the baby's brain development, and the baby's growth. Now we're gonna move to inheritance and we're gonna talk about genetics. So as you have already learned, the nuclei of all human cells, except gametes, they contain 23 pairs of chromosomes, right? The diploid number 2n, 2 times 23. One chromosome in each pair, it comes from the mother, and then the other one, it comes from the father. Each of these two homologous chromosomes contains genes that control the same exact traits. If one chromosome of the pair contains a gene for body hair, for example, its homologue will contain a gene for body hair in exactly the same position on that homologous chromosome. Alternative forms of a gene that code for the same trait and are at the same location on homologous chromosomes, those are called alleles. One allele of the previously mentioned body hair gene might code for coarse hair, and another allele might code for fine hair. Okay? Also, we have something called mutation. A mutation is a permanent heritable change in an allele that produces a different variant but of the same trait. Traits, phenotypes, and genotypes. So what is a trait? We have genetic and non-genetic traits. Okay, traits are characteristics. Some traits result from interaction of genes and the environment. Here we have some important terms. The word genotype. The genotype is the actual genetic makeup relating to a trait. The word dominant, so an allele that dominates or masks the presence of another allele is the dominant allele, right? And then the dominant alleles, we usually write it in an uppercase letter. As for the term recessive, the allele whose presence is completely masked is going to be the recessive allele, and then the recessive alleles are represented by a lowercase letter. And then another important term is phenotype. Just like the genotype is the actual genetic makeup relating to a trait, the phenotype is going to be the physical expression of the genotype. So the chart shows the possible combinations of gametes from two parents. So here, this is parent 1, and here we have parent 2, who each have one capital P and one little p. Okay, and then here we have the chart. This chart is called the Punnett square. So whenever we're constructing a Punnett square, the possible paternal alleles in sperm, they're written on the left side. Okay, and then the possible maternal alleles or in ova or the secondary oocytes, they're going to be written on the top of the chart. And then here, the 4 spaces in the chart show the alleles can combine in zygotes formed by the union of these sperm and ova to produce the 3 different combinations of genes or the genotypes, which we have the big P, big P, little p, and little p. So notice from the Punnett square that 25% of the offspring they have a genotype of big P, okay, and then 50% have the genotype of big P and little p, okay, and the rest of the 25% is gonna be little p. So people who inherit the big P and big P, little p, they're not gonna have PKU. And then those with little p genotype, they're gonna suffer from the PKU disorder. Okay, so PKU is the birth defect that causes an amino acid called phenylalanine to build up in the body. And then if it's left untreated, it can lead to brain damage, intellectual disabilities, behavioral symptoms, or even seizures. So although people with a big P, little p genotype, they have one PKU allele, the little p, right? The allele that codes for the normal trait, big P, so the capital P is going to be for normal, right? And then the small letter p is going to be for the PKU disease, okay? So the PKU allele. So the allele that codes for the normal trait, the big P, it masks the presence of the PKU allele, right? So an allele that dominates or masks the presence of another allele and is fully expressed, right? The big P in this example is said to be the dominant allele, right? So here in this case, this big P is going to be the dominant allele and then is masking the little p, which is the trait for the PKU. So those individuals will be normal, will not have the PKU disease. Also, the allele whose presence is completely masked, little p, right? You see how the little p is completely masked by the big P. Those are said to be recessive alleles, and the trait it controls is called a recessive trait. So by tradition, the symbols for genes are written in italics, with dominant alleles written in capital letters and recessive alleles in lowercase letters. So a person with the same alleles on homologous chromosomes, for example, big P, or little p, okay? So like this, big P, or little p, those are gonna be homozygous dominant or homozygous recessive. This is going to be homozygous dominant. This is going to be homozygous recessive. An individual with different alleles on homologous chromosomes, for example, big P and little p, okay, those are going to be heterozygous for the trait. And then again, what is the phenotype? The phenotype refers to how the genetic makeup is expressed in the body. It is the physical or outward expression of a gene. So a person with big P, little p, which is a heterozygote, has a different genotype from a person with a big P, which is a homozygote, but both have the same phenotype, right? So like this one is big P, this one is big P, little p, right? They have two different genotypes, but both their phenotypes say that the individual is normal, okay? So an individual with different alleles on homologous chromosomes, big P, little p, and then here, like we said, this is going to be the heterozygous. Okay, so heterozygous individuals who carry a recessive gene but do not express it, then they can pass the gene to their offspring, right? So like both those parents, they have big P and little p, right? And then they're both normal, right? Because they both have the big P, which is the dominant allele, but they can pass those little p's to their offspring, which is this 25% of the offspring. Those are gonna be the ones that they're gonna have the PKU disease. So nuclei of all human cells except gametes, they contain 23 pairs of chromosomes. Those are called diploid. They have 2 ends. Okay, gametes, they contain 23 chromosomes, haploid, or 1 end. The gametes are going to be the sex cells, okay? 1 chromosome from each pair came from father, and then the other member, it came from the mother. And then each chromosome contains homologous genes for the same trait. So genes are units of information about a specific trait, and each gene has a unique location somewhere in the DNA. We call it the loci, okay? And then there may be many different versions of alleles of a gene, and there may be many loci for a given trait. For example, like if this is your chromosome, let's say, this is a loci, this is a loci, this is a loci, and then on each loci, they will have a gene, right? And then with the homologous chromosome here, you will have the same gene with different versions of allele. Variations on dominant recessive inheritance. So simple dominance recessive is going to be dominant allele covers or inhibits the effect of the recessive allele, right? It masks it. So most patterns of inheritance do not conform to the simple dominant recessive inheritance we just described, in which only dominant and recessive allele interact. The phenotypic expression of a particular gene may be influenced not only by which alleles are present, but also by other genes and by the environment. Most inherited traits are influenced by more than one gene, and to complicate matters, most genes can influence more than one trait. So variations on dominant recessive inheritance include incomplete dominance and multiple allele inheritance. Okay, so first let's talk about the incomplete dominance and codominance. Okay, so in incomplete dominance, neither member of a pair of alleles is dominant over the other. And the heterozygote has a phenotype intermediate between the homozygous dominant and the homozygous recessive phenotype. An example of incomplete dominance in humans is the inheritance of the sickle cell disease, which we're gonna talk about it in the next slide. And then another variation of dominant recessive inheritance, we have the multiple alleles inheritance or multiple genes inheritance. So here, although a single individual inherits only 2 alleles for each gene, some genes may have more than 2 alternative forms. That is the basis for multiple allele inheritance. So one example of multiple allele inheritance is going to be the inheritance of the ABO blood group. Okay, so sickle cell is a group of disorders that cause red blood cells to become misshapen and break down. So people with the homozygous dominant genotype, okay, so here, which is the first one, okay, so HB just is for hemoglobin, but we're looking here, capital A, those are gonna have normal hemoglobin. And then those with homozygous recessive genotype, okay, so big S, big letter S, those are gonna have the sickle cell disease and severe anemia. And then although they are usually healthy, those with heterozygous genotype, okay, capital A and capital S, heterozygous because they're not the same, they're different, right? They're gonna have minor problems. So with anemia, because half of their hemoglobin is gonna be normal and then half of their hemoglobin is gonna be abnormal. Okay, so here in this case, heterozygotes are carriers and they are said to have a sickle cell trait. Okay, so here, as we can see, we have the 2 parents, okay, and then both parents are have the heterozygous genotypes. Okay, it means that they have the sickle cell allele, okay, and then it means that they have the sickle cell trait, okay, and then as you can see, once they have kids, so like let's say this is the sperm and then this is the ova, the maternal possible genotypes, and then here we can see that we have 25% of the offsprings are going to be normal, 25% are going to be normal, right, and then the 50% are going to be carriers, okay, just like the parents, okay, and then the remaining of 25% are gonna have the sickle cell disease. Multiple allele inheritance. Multiple allele inheritance occurs when genes have more than 2 alternative forms. So inheritance of the ABO blood group is an example of a multiple allele inheritance. So within this inheritance pattern, there is also codominance. Okay, so codominance means that 2 genes like type A and type B, for example, if we're talking about the blood group, those are both dominant and then they're both are expressed equally. So the 4 blood types, phenotypes of the ABO group, so we can either have blood type A, blood type B, AB, or the individual can have a blood type O, right? So those result from the inheritance of 6 combinations of 3 different alleles of a single cell called the I gene. Okay, so here this is the I gene. Okay, so for example, the first one, allele A, capital A. Okay, so like whenever you have AA or A and the letter I, okay, the person is gonna produce the A antigen. It means that that person has the blood type A, right? And then whenever we have the allele IB, that individual is gonna produce the B antigen. So like if you have big B or big B and little I, the phenotype of the individual is gonna be blood type B. And then allele I, it produces neither A or B. Okay, so the person's phenotype is gonna be blood type 0, right? And then whenever you have IA and IB, right? A and B are codominant, and then we said that both genes are going to be expressed equally, right? So the person is going to have both the A antigen and the B antigen, right, in its blood. So its blood type is going to be AB, okay? Notice that both IA and IB, they're inherited as dominant alleles, and then little I is going to be inherited as a recessive allele, right? And then because an individual with type AB blood has characteristics of both type A and type B red blood cells expressed in the phenotypes, so alleles IA and IB, those are said to be what? Codominant, okay? So in other words, both genes are expressed equally in the heterozygote. So depending on the parental blood types, different offspring may have blood types different from each other. So occasionally an error in cell division is called non-disjunction, okay? So non-disjunction, it results in an abnormal number of chromosomes. In this situation, homologous chromosomes, let's say during meiosis 1 or sister chromatids during anaphase of mitosis or meiosis 2, they fail to separate properly, okay? That's when we have non-disjunction. Also, a cell from which one or more chromosomes has been added or deleted, those are called aneuploid. Okay, so here we have a monosomic cell, okay, which is 2n minus 1 is going to be a chromosome set that is missing one chromosome. And then we have a trisomic cell, which is 2n plus 1. This is when we have an additional extra chromosome in our chromosome set, okay? So most cases of Down syndrome are aneuploid disorders in which there is trisomy of 21, okay? And then it means that on the 21st chromosome, we have 3 sets of our chromosome 21. So non-disjunction usually occurs during gametogenesis, which is meiosis, but about 2% of Down syndrome cases, they result from non-disjunction during the mitotic divisions in early embryonic development. Complex inheritance. Most inherited traits are not controlled by one gene, but instead by the combined effects of 2 or more genes. So that's a situation referred to as polygenic inheritance. Or it can be a combined effect of many genes and environmental factor is situation which we call is referred to as complex inheritance. Examples of complex traits include skin color, hair color, height, metabolism rate, and body fluid. In complex inheritance, one genotype can have many possible phenotypes depending on the environment, or one phenotype can include many possible genotypes. So for example, of an environmental factor is even though a person inherits several genes for tallness, for being tall, full height potential may not be reached due to environmental factors such as disease or malnutrition during the growth years. So skin color is a complex trait. Often, a complex trait shows a continuous gradation of small differences between extremes among individuals. It is relatively easy to predict the risk of passing on an undesirable trait that is due to a single dominant or recessive gene, but it is very difficult to make this prediction when the trait is complex. Such traits are difficult to follow in a family because the range of variation is large. The number of different genes involved usually are not known, and the impact of environmental factors may be incompletely understood. So skin color here, as we can see in this slide, is a good example of a complex trait. It depends on environmental factors such as sun exposure and nutrition, as well as on several genes. So suppose that skin color is controlled by 3 separate genes, each having 2 alleles. Okay, so it can be big A, letter A, big B, letter B, or big C or letter C, okay? So here, as we can see in this figure. So a person with the genotype, the first one, big A, big B, and then big C is gonna have very dark skinned, right? And then an individual with the genotype letter A, letter B, letter C is gonna be very light skinned, right? And then a person with the genotype big A, letter A, big B, letter B, big C, letter C has an intermediate skin color. Both those have intermediate skin colors. So parents having an intermediate skin color may have children with very light, very dark, or intermediate skin color. Okay, so here note that the P generation here is going to be the parental generation. Okay, is the starting generation. Okay, this is where it all starts. And then the F1 generation is going to be the first generation, okay, of the offspring, okay, and then is produced from the P generation, okay. So those parent 1 and parent 2, they're gonna have this offspring, right? And then we have, so this is F1 generation, and then here in this Punnett square we can see the F2 generation, okay, and then possible F2 generation offsprings, and then so those are going to be produced from the F1 generation, right? Autosomes, sex chromosomes, and sex determination. So when viewed under a microscope, the 46 human chromosomes in a normal somatic cell can be identified by their size, they can be identified by their shape and staining pattern to be members of the 23 different pairs. An entire set of chromosomes arranged in decreasing order of size and according to the position of the centromere is called karyotype, okay? Just like we can see here in this picture, this is a karyotype. So in 22 pairs, in 22 of the pairs, okay, so from number 1 all the way till number 22, okay, the homologous chromosomes, they look alike and they have the same appearance in both males and females, okay, like we have two of similar number 2, similar number 3s, similar number 4s, two chromosomes they look alike. So these 22 pairs, we call those are called autosomes, okay? And then the two members of the 23rd pair, okay, so this is the 23rd pair, those are these sex chromosomes, okay? Sex chromosomes. So they look different in males and females. So in females, the pair consists of two chromosomes called X chromosomes, right? The female is gonna have two X chromosomes. And then one X chromosome is also present in male, and then but its mate is a much smaller chromosome, which is the Y chromosome. Okay, so the female is gonna be XX, and then the male is gonna have XY. Sex determination. So when a spermatocyte undergoes meiosis to reduce its chromosome number, it gives rise to two sperm that contain an X chromosome and two sperm that contain a Y chromosome. So, and then oocytes, they have no Y chromosome and they produce only X containing gametes. Okay, so here, for example, sex chromosomes of the father, it can be either X or Y. And then sex chromosomes of the mother, it can be either X or X, right? So here we can see the father's possible sperm types, and then here we have the maternal ova types, okay? So, and then also one more thing, if the secondary oocyte, as we can see here, is fertilized by an X-bearing sperm, okay? Like for example here, one of the sperm has X and the other one has Y, okay? The offspring normally is female, XX, right? And then here we have XX, and then here we have XX. So fertilization by a Y-bearing chromosome, okay, sperm, it's gonna produce a male which is XY, XY. Okay, so here in this case 50% of the kids are gonna be females and then 50% is gonna be male. Okay, so here we can see that the individual sex is determined by the father's chromosomes. So both female and male embryos, they develop identically until about 7 weeks after fertilization. At that point, one or more genes set into motion a cascade of events that leads to the development of a male. So in the absence of normal expression of the gene or genes, the female pattern of development occurs. Just a fact on the side, so it has been known since the 1959 that the Y chromosome is needed to initiate the male development. So now we're going to talk about sex-linked inheritance. So in addition to determining the sex of the offspring, the sex chromosomes are responsible for the transmission of several non-sexual traits. Many of the genes for these traits are present on X chromosomes but are absent from Y chromosomes. This feature produces a pattern of heredity called sex-linked inheritance that is different from the patterns already described. Okay, so for example, we have the red-green color blindness, which is one example of a sex-linked inheritance, the red-green color blindness. This is the most common type of color blindness disease. Okay, this condition is characterized by a deficiency in either red or green sensitive cones. So red and green are seen as the same color, either red or green, depending on which cone is present. So the gene for red-green color blindness is a recessive gene and then is designated in small letter C, okay? And then it's gonna be small letter C and then the normal color vision, okay? So this is gonna be color blind, and then for the normal is gonna be which dominates, okay, it's gonna have the capital C, and then that one is for normal. So the big C, little c genes, they're located only on the X chromosome. So the ability to see colors depends entirely on the X chromosome, okay? Like here, as we can see in those examples, the Y chromosome, it doesn't have any, is not carrying any Cs, okay, any alleles. So the possible combinations are as follows. For example, if we have a genotype of big C and big C, right, the phenotype of the individual is gonna be normal and the individual is gonna be female because it has two X's. And then if the genotype is big C and little c and the individual is female, the individual is gonna be normal, but is gonna be a carrier of the color blindness, right, for the recessive gene. And then if we have a little c for a female individual, that female individual is going to be colorblind because it has two of the recessive genes, right? And then here we have capital C. So this individual is going to be a male because it has a Y, and then because it has the capital C, the male is going to be a normal male. And then here we have a little c and the individual is a male. So here, unfortunately, as you can see, the male, even though it only has one C, the male is gonna have is gonna be a color blind male, okay? And then as for females, okay, as you saw here, that the female needs to have both letter C's to become colorblind, and then if it only has one, then the female is normal but is a carrier for the genes, okay? So here we have a Punnett square for the inheritance of red-green blindness, and then just like we said, the normal individual is gonna have the big C, right? And then once you're a female and then you have two little C's, you're gonna be colorblind. Or if you're a male and then you have one C, you're gonna be colorblind, okay? So here we have the male, And then this male has capital C, so it's a normal male, and then there's nothing on the Y chromosome. Here we have the female. So the female is a carrier because it's also normal because it has the capital C, but it's carrying the lowercase c, right? So here we have the possible sperm types, and then here we have the possible ova types, and then as we can see here we have 25% of the females, they are normal females, and then we have 25% of the females are gonna be carriers because they have the big C and a little c here. And then 25% of the males are gonna be normal males, right? Because they have the capital C. And then here, as you can see, 25% of the male are gonna be colorblind because they have that one letter C there, okay? So this is the end of chapter 29, Development and Inheritance. Please let me know if you have any questions. Thank you.